page, and remember, pause this whenever you need to. This is going to be the last part of the notes for Unit 10 Biochemistry. So the last little bit here, and we're doing B9C, Part 2. And what this again says, it's really short, but it's got a lot of information in it. It says identify and investigate the role of enzymes. So ID, identify, and investigate the role of enzymes. So we did carbohydrates, lipids, proteins, nucleic acids, and now we're hitting on proteins again because enzymes are proteins. And when I say role, someone asked me earlier what I was talking about here. Think of it as their job. What job do they have? What's their role? What's their function? What do they do here? And one thing I'm going to tell you first off, and I'm just going to write this word over here. Many of the questions on the STAR test with this particular TEK are going to be in graph form. They're going to have some graphs. So I'm going to show you some graphs here when we're finished with this, and they're going to be just all over the place. Graphs, graphs, graphs. That's what they're... I pulled out, I think, 5 out of the questions they released had graphs on them. The other ones had like a little flow chart, and I'll talk those, show you some of those tomorrow. So investigate means that's what our lab is on Wednesday. So on Wednesday, we're going to use pectinase. If it has ASE, it's an enzyme on the end of it, ASE, so pectinase. And pectinase is what's used to make apple juice. So we're going to try and make apple juice without and with the enzyme. So with the enzyme and without the enzyme, we're going to see how much we can make and see what happens faster. Okay? So when we deal with enzymes, there are some problems that are just naturally associated with these. These are big, huge Godzilla chemicals, but they're really uber sensitive. So enzymes, remember enzymes are proteins. Sorry, let me put that in parentheses. These are proteins and they can be easily denatured. So enzymes can be easily, having problems writing now. Denatured. And denatured means you mess up the shape. So it's messed up, or you have an altered shape. And if you mess up the shape, it doesn't work right anymore. Remember how, I'm going to flip this over one page. Do you remember how before spring break we talked about the fact that here's your enzyme, and it has a certain shape in it that fits chemicals in it, right? If the shape is not right, it's not going to function right anymore. So the shapes are really important. Now, I gave you really simplified shapes like this. The shapes are a lot more complex than that, but that gives you a pretty simple idea of what's going on here. So it is easy to denature an enzyme, and you can denature it by messing up, the pH. So the pH is talking about whether it's an acid or a base. Okay, and the pH scale goes from 0 to 14. And 0, closer you get to 0, it's more acidic, and the closer you get to 14, it's more of a base. And 7 is neutral. You guys have heard of that before. If you've ever tested the pH of your pool or a pool somewhere, it should be, I think it's a little bit above 7, a little bit basic, I think is where it's supposed to be in the range. Yes, am I wrong on that? No? I think it's supposed to be a little closer to 8 here. We're most familiar with acids. The vast majority of foods that we eat are slightly acidic. Drinks that you drink, lemon juice, lemonade, orange juice, sodas all have an acidic layer to them. So the closer it is to 0, so when you have a low pH, that's acidic. When you have a high pH, that's basic. Okay? So different enzymes work best at different pHs. It depends on the chemical what pH it works best at. So for example, you have enzymes in your stomach. Would they work at a high pH or at a lower pH? In your stomach. Why? Why do you say lower? Yeah, you have that acid in your stomach, right? That makes sense. There are some enzymes that if it's got the stomach acid in it, if it's they won't work at all. Not at all. Your amylase, if you're drinking Dr. Pepper or Coke while you're eating something starchy, like fries, your amylase doesn't work very well because the pH gets too high for it, or I'm sorry, too low for it, too acidic for it. And so it's best to drink water when you eat because that allows the amylase to work and starts that digestive process. Less indigestion. You guys, sometimes you eat so fast you get heartburn, stuff like that. Yeah, especially with your shortened lunch, you do that. So that's one of the little things. Drink water instead of soda with it. Okay, these enzymes are very sensitive. They actually, strangely, are easily messed up. Let me see if I can make this a little bit bigger. That might be too big. Okay, so pH is the first one. The second one is temperature. We can mess them up with temperature. So it's the same with temp. So I'm not going to write all that stuff again. It's the same with temperature. If your temperature is too high or too low, it's not going to work right. When animals hibernate in the wintertime, everything slows down, their body temperature lowers, and their enzymes don't work as well anymore. That's one of the ways that they can slow things down. Our temperature, our enzymes work best at about 98 degrees, right? That's your body temperature. If you were a fish, it would have to be able to work over a greater range. So there might be, because the fish are cold-blooded, right? Fish, reptiles, amphibians, they have differences in their body temperature. Like their body temperature can lower with the air around them or it can increase with the air around them. But there's a range. If it gets too cold, those fish are going to go into stasis, kind of like a stasis, right? Like a hibernation. If it gets too hot, a similar thing happens. Especially with reptiles and amphibians, when it gets too hot, like let's say it's 100 degrees outside, which we hit every year, right? If it gets too hot, their bodies, their enzymes can't function at that high temperature, so they'll go den up. So the heat of the day, the hottest part of the day, a lot of times you won't see reptiles and amphibians laying around. They'll be in a cooler area in the shade. Sometimes it gets so hot that they'll even estivate, which means they lower their body processes slower, so everything is kind of like slowing down for them. They'll be a little bit sluggish. That doesn't mean stick your hand in with the rattlers while it's, at 4 o'clock where it's 102 degrees outside. Don't do that. They're still active. It's just they're not going to move as fast. Fast enough to bite you, but still not as fast as normal. Every year some idiot finds a bear den, and they go in while they're hibernating and thinking, oh, they will wake up. No. Hibernating does not mean that they're in a coma. It means everything's slowed down, so they won't react as quickly, but they will wake up and kill you. Yeah? So hibernating and estivating, opposite ends of the temperature scales there. So the same thing's going to happen with temperature. And again, let me give this example, because I think this example is one that every single one of you guys will have access to. Detergents, if a detergent has an enzyme in it, and they do have enzymes because it helps them to clean stuff better, the detergent with enzymes works better in cold or warm water. Not in hot water. Because what happens is you get the water too hot and the temperature breaks down the structure of the enzyme where it's not going to function anymore. It doesn't work anymore at all. It won't be able to break down that ketchup stain that's on your shirt.
You didn't tell us to say hi to
Oh, I'm sorry. Say hello to everybody online.
Hello.
The next one is a weird one. It's called salinity. Anybody know what that one is? You might have heard the word salinity, salt content. Same thing happens with salt content. It works best at a certain salt content, salinity. So a lot of times we talk about this in terms of animals that live in the ocean compared to animals that live in fresh water. Yeah, they have to worry. They really have to manage the salt content in their bodies. If they're living in fresh water, they're constantly having to get rid of water and keep the salt content in their bodies. If they live in salt water, they're constantly getting rid of salt and trying to keep the water inside their bodies. So they really have to work hard at it in order to do that. Humans not so, ours is, you guys have tasted your sweat, you've tasted your tears before, you know that's got a salty flavor to it. Yes? So you know we have a lot of salt in our bodies, but it's much easier for us to maintain that than it is for organisms that live in different water environments there. So salinity is a thing. Too much salt or too little salt is going to mess up how the enzyme functions. And then there are these things called inhibitors. How much time do I have left? Oh, I'm good. Okay. Inhibitors. That one sounds really young. Okay, so inhibitors are chemicals that stop the substrate from attaching to the enzyme. If you look at the last page of notes, we talked about the substrate before. It stops the substrate from attaching to the enzyme. Okay, I'm going to flip over a page real quick and I'll bring it right back here. So when we talk about the enzyme, okay, so remember here's the enzyme, right? And the chemicals that attach to it so they can chemically react together or break apart, those are the substrates. Sometimes there are chemicals that fit into this, so something's in here and so this chemical substrate number 2 can't fit. It's already blocking the space. Good and bad. Some snake venoms will work like that. Well, they'll get in here in this active site, so this other chemical can't get in there. And a lot of times it's something that's really, really important in order for you to live, and so you die pretty quickly from that. Sometimes we use them in a good way. Like think about aspirin, Tylenol, things like that. That's how they work. Okay, so let's say you have a headache. Okay, you have a headache. Oh, let's do something different. Let's say you sprained your ankle. You're walking up the steps, I don't know, your buddy pushed you, whatever, you sprained your ankle and it hurts really bad. So you take some ibuprofen, okay? And then after about 15 minutes, it doesn't hurt nearly as bad. Did something heal in those 15 minutes? Nothing healed, nothing changed. The only thing that changed is you stuck a chemical in there that inhibited how your neurons talked to each other. Neurons, nerve cells. So when your ankle neurons, what they do is they send this pain message up your neuron, and then there's a gap, and there's the next neuron, and there's a chemical that crosses the gap. Well, when you take ibuprofen or Tylenol, there's an inhibitor that stops that chemical from chemically reacting and crossing the gap. So the chemical can't get the message from one neuron to the other. So the pain message in your ankle never makes it to your brain. It's not that there's not pain there, it's that your brain never knows. That's how a lot of pain medications work. They cut off the message to your brain that says, hey, that hurts. It works, doesn't it? Now, the bad thing about that is if it stops hurting, what are you going to do? You're going to walk on it and maybe damage it a little bit more, so you have to be careful with pain medications. But the good thing is, a lot of times when you take them for things like headaches, one of the reasons you have headaches is because of stress, right? Stress headache. You ever have someone rub your shoulders and it makes the headaches feel a lot better? Because usually when we have a headache, we tense up our shoulder muscles here, and as it hurts more, you get more and more tense, and so it kind of exacerbates the problem. When you take pain medication, it doesn't go just to your ankle, it goes everywhere. And so now the message that your neck and your head's hurting never makes it inside to the brain. It cuts down a lot of those messages, and so you don't know. And a lot of times that totally makes the headache go away, because once you don't feel the pain anymore, don't you relax? Yeah, and it helps it to go away.
It's amazing how you can physically think about being in pain and also not be in pain.
Yes, yes. Yeah. Okay, so I'm going to flip back a page. So inhibitors, they can be really bad for us. They can also be really good for us. And your body produces some just automatically too, because sometimes it wants the reactions to happen, sometimes it wants them to stop. So it can keep it from going, it can inhibit the enzymes from reacting that way too. Okay, examples. Digestive enzymes. We have a lot of digestive enzymes. And again, some of those help digest carbohydrates, some of them help digest proteins, things like that. And for us, for this particular type of enzyme, it's actually breaking stuff down, right? You get the starch and it's going to break it down into simple sugars. You get the protein, it's going to break it down to amino acids, right? So it helps break down, digest. That's break. Carbs, proteins, lipids, and so on. These are examples like amylase. That's the stuff in your mouth. Remember, we had a little example of that one. What do you think protease breaks down? Yeah. What do you think lipase breaks down? Yeah. Yeah, so in many cases, the name helps us figure out what it breaks down. Not always, but in many cases it does. A lot of times they're named after the chemical that they help to break down. Sometimes it's a big generality, sometimes it's really specific, and you're thinking, crap, I don't even know the name for that one there at all. We also have some metabolic enzymes. We have lots of enzymes everywhere. Metabolic enzymes, and I'm going to bring these two up because we're going to talk about them in the next unit. DNA helicase. Okay, what do you think it breaks apart? The DNA what? Helix. Helicase, it's going to break apart it's kind of like unzipping parts of it. So this DNA helicase breaks open the ladder. And then another one is DNA polyrite polymerase. This one is an enzyme because it's got ASE on the end of it, but it actually builds stuff. It doesn't break anything apart. This is what happens. You know how through mitosis you have to make a copy of each of the DNA strands? Yes, your new cells can have it. This is what does that. So this adds nucleotides. Remember, they're the basic unit of the DNA structure or the RNA structure. Adds nucleotides to the new DNA. So it helps to build the DNA. Okay, once you've written that down, I have two questions I want to show you, and then you can pack it up, okay? The first is this one. Ignore all the writing. Don't even worry about the writing. Can you tell what this graph is about?
pH.
Okay?
Because the neutral is 7, so pH.
Okay, so you've got pH, it's got neutral, most acidic, most basic. How many enzymes are on here? Two of them. Look at this. We have pepsin, we have trypsin. Do they both work best at the same pH?
No.
No. Which one works at the most acidic pH? Where in your body is it working? Why?
Because
That's where the acid is. That's exactly what they're asking you here. Now look at this one. Sit down. We're almost there. You have time. You're not going to miss the bell. Look at this one now. This one has some horrible stuff. Here's that, TEK polymerase and so on. But look at the graph. Notice it's temperature related, right? Okay. If I said that the enzyme works better as the temperature increases, is that a good thing to say? No, it doesn't always. If I increase the temperature to 90 degrees, is it still working better? No, it's not at all. You have to limit yourself to what the graph has here. Which statement about the enzyme activity is best supported by this graph? Okay. An enzyme must be composed of multiple polypeptides or subunits to be active. You know what? That's true. But is that what the graph is telling you? No. An enzyme's rate of activity increases with time until it becomes inactive. Does it just automatically increase with time? No. An enzyme functions best under specific temperature conditions. There you go. Okay? Go ahead and pack it up. Okay, I'll see you guys tomorrow.